everyone, welcome in again in our laboratory. From the previous video, we talked about atomic spectroscopy and its subtopics. But now we will move on to mass spectroscopy. Before that, let me present our learning objectives and our conceptual framework. And now, let's begin with the discussion.
Good morning, everyone. Today, we will be learning about what is mass spectroscopy. So, mass spectrometry, or also called mass spectroscopy, is an analytic technique by which chemical substances are identified by the sorting of gaseous ions in electric and magnetic fields according to their mass-to-charge ratios. So, the foundation of mass spectroscopy was laid in 1898 when Wilhelm Wien, a German physicist, discovered the beams of the charged particles could be deflected by a magnetic field. In more refined experiments carried out between 1907 and 1913, the British physicist J.J. Thomson, who had already discovered the electron and observed its deflection by an electric field, passed a beam of positively charged ions through a combination of electroelastic and magnetic field. The two fields in Thomson's tube were situated so that the ions were deflected through small angles in two perpendicular directions. The net result was the ions produced a series of parabolic curves in or on a photographic plate placed in their paths. This is the reason why J.J. Thomson credited with the construction of first mass spectrograph and first mass spectrometer. The history of mass spectrometry or mass spectroscopy has its roots in physical and chemical studies regarding the nature of matter. The study of gas discharges in the mid-19th century led to the discovery of anode and cathode rays, which turns out to be positive ions and electrons. Improved capabilities in the separation of these positive ions enable the discovery of stable isotopes of the elements. The first such discovery was with the element neon, which was shown by mass spectrometry to have at least two stable isotopes. We have 20 Ne, or neon with 10 protons and 10 neutrons, and also 22 Ne, or the neon with 10 protons and 12 neutrons. So we will be moving on to the types of mass spectroscopy. So mass spectroscopy is a powerful analytical tool with high sensitivity and high mass accuracy. Recent technical innovation in mass spectroscopy-based techniques have contributed to a range of highly sensitive and versatile instruments. So the first is the matrix-assisted laser desorption ionization time-of-flight mass spectrometry is commonly used. It is a soft ionization technique to create ions with minimal fragmentation by using a laser energy. In time of flight, the protonated ions are accelerated by an electric field to make an ion have the same kinetic energy as any other ions or ions that have the same charges. The second one is what we call the triple-quadruple mass spectrometry. So it is a tandem mass spectrometer in which the first and the third quadruples act as mass filters and the second acts as collision cell, fragment the selected precursors, parents ions, and to generate fragment or daughter ions. So the third would be the quadruple trap mass spectrometry because it is a hybrid. This instrument retains the classical triple quadruple scan function and it is used for sensitive ion trap experiment. The fourth one is the hybrid linear ion trap orbitrap mass spectrometry. It combines a linear ion trap and high resolution orbitrap. So the last but not the least would be the quadruple orbitrap mass spectrometry combines high performance quadruple precursor selection with high resolution. It can be widely used in various fields like proteomics, metabolomics, food safety, toxicology, and so on.
So now let's discuss about the basic principle of mass spectroscopy. So mass spectrometry generates multiple ions from the sample under investigation. It then separates them according to their specific mass to charge ratio, and then records the relative abundance of each ion type. So now let's discuss about the application of mass spectroscopy. So mass spectrometry is an efficient method to elucidate the chemical composition of a sample or molecule. More recently, it has been used to classify biological products, in particular proteins and protein complexes, and a number of species. Usually, mass spectrometers can be used to classify unknown substances by molecular weight measurement, to measure known compounds, and to determine the structure and chemical properties of molecules. Due to its capability to distinguish between substances, mass spectrometry is used to determine the unknown substances, and also to identify the isotopes of a substance. In analytical laboratories that study the chemical, physical, and biological properties of a substances, it is favored over several other analytical techniques, as it has less background interference since it is performed in a vacuum.
Now let's proceed with the instrumentation and diagram. There are four main parts of mass spectrometry, which are the ionizer, accelerator, deflector, and detector. First is the ionizer, which is the bombarding of the sample is done by the electrons. These electrons move between cathode and anode. When the sample passes through the electron stream between the cathode and anode, electrons with high energy knock electrons out of the sample and form ions. Second is the accelerator. The ions placed between a set of changed parallel plates get attracted to one plate and repel from the other plate. The acceleration speed can be controlled by adjusting the charge on the plates. Third is the deflector. Magnetic field deflects ions based on its charge and mass. If an ion is heavy and has two or more positive charges, then it is least detected. If an ion is light or has one positive charge, then it is deflected the most. Fourth is the detector. The ions with correct charge and mass move to the detector. The ratio of mass to charge is analyzed throughout the ion that hits the detector. So how does mass spectrometry work? In a regular mass spectrometer, we initially have the material to be analyzed, but we need it to be ionized to pass through the spectrometer with enough energy. Thus, the sample is bombarded by electrons to ionize it. The ionized beam is now passed through a series of electric or magnetic fields depending on the type of the sample and its properties. The ions are deflected by the field throughout in such a way that ions with the same mass to signal ratio will follow the sample path to detector. These charged and deflected ions are now incident into a detector which is capable of distinguishing the charged particles falling on it. Based on the mass spectrum produced by the charged mass, he can identify the atoms or molecules constituting the sample by comparing them with known masses or through a characteristic fragmentation pattern. We will now proceed with spectroscopic techniques of mass spectroscopy. These are some guidelines or methods to ionize samples. First is the electrospray ionization or the ESI. The sample ionized by ESI are introduced into the instrument in a solution of polar solvent via spray at atmospheric pressure. It is the softest ionization technique available and produces protonated or cationized molecules in the positive mode and the protonated molecules in the negative mode. Second is the atmospheric pressure chemical ionization, or the APCI. It is similar to ESCI in the way samples are introduced and the type of ions formed, which is the protonated or deprotonated molecules. It is not a soft technique just like ESI. It uses a spray and thus requires some sample volatility. Third is the atmospheric pressure photoionization, or the APPI, that uses UV photons to ionize molecules and can form molecular ions or protonated molecules. It is more energetic ionization technique and ionizes sample that may not be efficiently ionized by APCI and ESI. Fourth is the liquid chromatography mass spectrometry, which combines the separation power of liquid chromatography with the ability of mass spectrometry to characterize compounds. The instrumentation system consists of a binary pump, an autosampler, a C18 reserved phase column and 2 detector, an UV detector, and mass spectrophotometer. The mass spectrometry only has ESI source available. This technique is applicable for some samples soluble in acrylonitrile, methanol, and water. Fifth and the last, which is the electron ionization, or the EI. EI is the original ionization technique and uses energetic electrons to produce molecular ions along with cluster fragment ions. EI is the most often coupled with mass chromatograph for analytical purposes. Remember that often samples may be ionized by more than one technique. If there is an advantage with mass spectroscopy, then there will also be a disadvantage. A big advantage of mass spectrometry is that it is incredibly sensitive over many other techniques. It is an excellent tool for identifying or confirming presence of unknown components in a sample. The disadvantage are that identifying hydrocarbons that produce similar ions is not very good, and it is not able to separate optical and geometric isomers.